Welcome everybody. I'm Tobias Hamp. Burkhard Rost is currently on travel in Brazil, so I will be giving this lecture and the one on Thursday. My topic will be the homology-based prediction of protein function for both lectures, but don't worry if you don't really understand the title yet. I will be giving a very extensive introduction, repeating some of the stuff you have heard last week, and giving you some new perspectives as well. So I'm just curious. So who of you already had predicting protein 1? None of you? Okay, good. Okay, as I said, let's start with an extensive introduction to the whole field in general. So we are in Garching here at a research cluster. Many faculties around us build tools and also tools to make life easier. So here you learn how to automate things with software. Next to us there are people who build machines that make you travel faster, or build materials that are light and very strong. And an interesting trend in research is generally that they are somehow converging towards our human body. So, for example, you have DARPA projects in the US which tries to build machines that act like animals and can carry very heavy weights. You have something like exoskeletons which try to help your arms and legs move. Or you have little microchips that you can implant in your brain that help you with certain neurological diseases. Or even software projects which try to, for example, emulate certain parts of animal brains, and even go even as far as trying to simulate the human brain, although this is very much at the beginning still. And so molecular biology in general and computational biology has a very different approach to all of this. So we are basically researching life on the molecular level. And if you spend a little time in the field, you will be asked by many people what exactly is computational biology, and this is usually the answer that people will remember the best. And the one follow-up question to this is usually that people are confused because they believe, at least for a human, that we already know everything about the molecular detail. So it is easy to get this impression, for example, when you're at a doctor who tries to somehow give you the feeling that he knows everything, that he's in control of what he does, or by media series like Dr. House, for example. But it is clearly not the case. So we do not understand life yet on the molecular level, and it's not even close. It is true that we have made enormous progress, so we know many treatments to various diseases. But if you would draw an analogy to the computer world, you would have basically bought a new PC and now tried to overlock the CPU a little bit, buy a new fan, exchange graphic cards, something like that. So you have no specification of the whole system, no idea how the chip really works, no idea about the code of the operating system, none of that. And this is really bad luck, because again, if you spend some time in the field, you get a feeling for the progress in general, and it's going really slow. And it's much slower than everything that is going on in digitalization. And again, in terms of molecular biology, so we are really not a very blessed generation. So maybe our grandchildren, grandchildren will be benefiting from all of this, but we probably not. And sometimes you get the feeling that some CEOs at certain companies in their quiet moments get the same impression and try to change that, because they realize that all the billions they have is not worth anything if they go to the doctor and the doctor says he has cancer or something. And Google just recently started a big project, but so far so many companies have tried to go this way, but also with very limited success. Okay, so what exactly is the problem on the molecular level? So again, if you compare various machines here on the top, they're all basically built from the same principles. So they all have microchips, displays, hard drives, graphic cards, and so on. So basically it's all the same. And if you would really want to know every detail, you could have it. So there is a specification for every single part in these machines. If you go to the human body, you sort of get the same impression. So 7 billion people on Earth, and they all built more or less in the same way. So same organs, same basic type of thinking. You could argue against that, but on an absolute scale, we are all very, very similar. And the big question here is why is this the case? So why can't we really look into this? So let's follow a top-down approach first. So this is what you all should have learned in school. So human body consists of various organs. Organ can be compartmentalized again into different subgroups, and if you go further and further down the scale, you end up with cells, which are composed of different organelles. For example, in pink here, the cell core. Again, in school, high school, you should have seen something like this. This is a bottom-up approach. So you go to the very most detailed levels, the DNA level, and you learn that DNA is transcribed into mRNA, and mRNA is again translated into proteins, which are basically the things that do something in the cell. Again, so if this is not familiar, I would strongly recommend you read up on it as soon as possible. If you put this picture together, so the top down on the right, bottom up on the left, and you simply compare the scales. So 1 nanometer means 10 hydrogen atoms in a row, basically. So the diameter of DNA is 10 hydrogen atoms in a row, so 10 hydrogen, so one nanometer. Proteins are a little bit bigger, but they are globular, so they are balls, basically. Then you go to the cellular level, and you suddenly have a different scaling here. So the average diameter of a cell, 10 micrometers. And if you do a little bit of math, you quickly realize that you can fit 100 billion proteins into one cell. So again, so I repeat. So proteins are, the average protein is small enough to fit 100 billion times into the average cell. So this is sort of a big step here. And in high school, typically, this part is totally left out. So you may learn about some complexes on the protein level, but how you get from this level here to that, so this is basically a plot hole like in a movie. And the reason for that is simply because we don't know. So we don't know how these proteins interact together in order to form such a complex thing. And this lecture is basically about this arrow right here. So what is the first step after the protein? What is the function of the protein? And over the years, we hope to finally to bring this whole picture together and be able to answer the question how these things form such a cell. Okay. Yeah. So this is a small movie to show you what we would ideally want to have. It's 3 minutes long about.
Display of choreography. All the components of a molecular machine locked together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
All right. Again, so this is what we ideally want to have. So this is one of the very few processes that we really understand on the molecular detail. And so the video stopped with a synthesized protein, namely hemoglobin. So this is not hemoglobin, but let's just say this is any generic protein. In the example, it is ATPase. So this protein, once it is formed, once it is folded, would then go on to attach itself to other proteins to form a complex, as you see here on the right. So this is an ATPase. So different colors indicate different proteins, and this machine here that you see is responsible for the synthesis of ATP in our body. And ATP is like an energy currency, and for example gives the energy for your muscles to move. And again, so this is one of the very few complexes where we really understand very much. So where the molecular details are all known. So and again, so every ball that you see here is one atom. Okay. Another big misperception often is the power of simulation. So many people assume that once you have the coordinates of every atom of any material, basically, and in particular of proteins, that you can now go on and simulate the behavior of this molecule on the molecular level for as long as you want and in as much detail as you want. And this is absolutely not the case, unfortunately, because reality is that even in such a simple grid that you see here, where every ball corresponds to one atom, so the influence of one atom influences all the others, and it is already computationally quite expensive to predict the behavior of every atom here over time. And we are talking here about a scale of picoseconds, which is a billionth of a microsecond, and a microsecond is a millionth of a normal second. And even this behavior here is already not quite accurate. So you would think that this is really the truth that you see here, but already this is simplified in a way that you have made some compromises towards truth. And the same is true for this simulation here. So this is, I believe, one microsecond, so one millionth of a second of a simulation of a protein. And this is basically so on this scale, all the molecular dynamic simulation are computed nowadays. So we cannot go to the level of seconds or even days or something. And if you compare this, for example, to the Ebola virus that is currently in the news with an incubation period of 21 days, so clearly there is not even a chance that we simulate anything like that. So you need something different. And yet another misperception is the power of gene manipulation and gene therapy. So you could argue that everything I say is somehow questionable because we already have successes with gene therapy and gene manipulation, and we can already do a lot there. But what people in this field are doing is basically they change some part of the code and see what comes out in the end, without really understanding what is going on in between. So it's like running a program and then changing a couple of bits in the heap memory and see what happens. So from time to time you hit something useful that you actually wanted to change, but most of the time you will do something that doesn't really make sense or is harmful to the program. Okay. Yeah, so this is a small test for you. So guess when this picture of a protein was taken. Any guesses?
1950s.
Okay. That's realistic, yeah.
Black and white.
Sorry?
I guess it's black and white.
Yeah, this doesn't, so it could be from an X-ray or something, right?
Ah, yeah.
Okay. But I guess the black and white, you immediately have some sort of judgment in your head. Okay, so this was 1965. Okay, one of the first. So this is actually not a direct photograph, but somebody built this protein with some sort of mass that is like this big, and then photographed it. Because he only had the coordinates of the atoms, and not so no software to really view it. And so 1965, so 50 years ago, and so human has like 20,000 proteins, and today we only know this level of details for about a quarter of them. So this gives you some imagination how slow things are progressing. So for the very most detailed levels, at least. And yeah, so going back to the first slide again. So I hope you have a better picture now. And yeah, and if you want to travel to the edge of human knowledge yourself, you can easily do this by just browsing Wikipedia. What I want to show you now. So, and I will do this for the biological process of sweating, because it is simply the most simple process I could think of. So heat comes in, water flows. So this is it. I couldn't think of anything simpler. And so oh no, oh, this is deadly. Okay. So, wait, let's go to Wikipedia. Turning your smartphones off might help. with my browsing. Okay, so perspiration, the scientific term for sweating. So here, perspiration is the process of fluids secreted by the sweat glands. What are sweat glands? Sweat glands, so are small tubular structures that basically carry the fluid. There are two types: eccrine sweat glands, apocrine sweat glands. We are interested in the molecular details of sweating. Obviously, you will have very little chance of getting the details for the first part. So for the part that is happening in your brain, because this is really a part where we have basically no idea about how this works, and you can somehow see this under the category mechanism, and then stimuli, thermal. So both types of sweat glands participate in sweating, controlled by the hypothalamus, and sweating is stimulated by a combination of internal body temperature and mean skin temperature. And I've seen papers recently as 2013 which tried to investigate how exactly body temperature and actually brain temperature influence the outcome of sweating. And so as you can imagine, if you're still at this level, there's absolutely no chance to get the molecular details of this. So let's just assume we skip this part, and we want to know what happens once the electrical signals from your brain reaches your skin. This is what is described here. So the eccrine sweat glands, those are the most common sweat glands. The stimulation occurs via activation by acetylcholine. So the acetylcholine, yeah. So this is the transmitter of the electrical signal, which binds to the muscarine receptors. So muscarine receptors, I go there. Okay, so I will give you information. So there are various types of muscarine receptors, and we are interested in the type M1 here. So you can read it up again at home, and so this is basically already a different name for a gene. Gene results in a protein. So let's follow this one. Now you're already at the DNA level. So you check out the protein that corresponds to this gene. So this is protein resources UniProt. So UniProt is the collection of everything we know about proteins, basically. And you're close, so you already have for this particular protein a number of functions, but you want to go even deeper and go to the structure. And what you have here are 3 databases, but I can tell you that none of them has really an experimental structure. So they're all predicted structures by computational biology. And the reliability of these is already questionable. So basically, this means this is the end of the line. We do not know how the receptor for this acetylchylins looks like, and therefore we are already somewhat limited for the molecular details, and this is it. So we cannot go any more detail for this most simple process called sweating. And yeah. Oh wait, too far. So why is that? So we clearly the devices that we have are not good enough yet. They are too slow. They make a lot of mistakes. So a lot of the data that you come across in molecular biology, you cannot immediately trust. So you need to repeat the experiments. Conduct different experiments to really have a definite answer what is going on. All this costs a lot of money, and research money is usually limited. And of course, computing. So a lot of answers you can answer by simulation. For example, what a protein looks like can be addressed with enough computing power. But also, this is obviously quite limited if you scale it up. Then some advices for you as computer scientists. So I've just covered the molecular biology part. Generally, in biology, there are exceptions to every rule. This is much unlike in computer science. So every statement that you make, every statement that you read in a book, there are exceptions to it. This means you will have to deal a lot with probability theory, especially if you do predictions, and you should be familiar with terms like the mean, the standard deviation, the standard error, the accuracy, the receiver operating curve, and the area under this curve. So I haven't written down the exact formulas, but you should be familiar with them. And yeah, so what computational biology does is really probabilistic abstractions of biology. So it tries to model proteins, for example, abstract proteins, and go on from there. This is much in contrast to basically anything that is going around us. So we don't really care how fast our program is, as long as it finishes within a year or something. We don't care if it crashes for different input. We just care for our input. We don't care if it's safe. It is if people can crack it. If it easily usable, at least as long as we are doing the research ourselves. Documentation, obviously, also not as important. The way you write the code, also not so important, as long as the code runs. What really matters is how good is your probabilistic model. So how good are your predictions to answer biological questions? Okay, and this brings me directly to the actual topic of today's lecture, namely the homology-based prediction of protein function. Last week, what was covered? So I have repeated a few things, given you some new perspectives. Again, you should really read up on this. So what is a protein here on the left? An atomic view, on the right a more abstract view, which is not as accurate, but gives you an impression that you can handle better in the end. The data in molecular biology grows faster than any CPU or memory, so known sequences grow extremely fast, and so basic information about what they are doing, some simple annotations, publications, and so on. You have heard that many proteins are enzymes, actually. So enzymes catalyze certain chemical reactions. And you also have seen today and also last week some simple molecular dynamics simulations. and also you have heard about protein disorder. So not every protein adopts a defined structure on its own. So a disordered protein is something that you hear on the left. So this is the unfolded chain, and only upon contact with an actual folded protein, this disordered structure adopts a definite form that finally that actually does something to the protein it binds to. All right. So now something totally different. So this lab year started in 29, I believe. So 2010 was some of our first lecture, and as I said, so the molecular details of many proteins are still missing today. There's not a chance that we close this gap anytime soon. Yet we do have a lot of sequences of proteins. So the human genome is completely sequenced, and we have sequences from many other organisms, but we don't know what these sequences do. And one very simple way of learning what they do is to take an unknown sequence and to run it against a database of known sequences for which we already know something. And the idea for this exercise in predict protein at this time was exactly that students implement simple programs which do exactly that. So we had 16 students at the time. They were split into 3 groups, and each group was supposed to implement their own predictor. And so you were given sequences and predict functions, and now you had to somehow define what is a protein function. So what is it that we actually want to predict? And Bokatros himself said a while ago, function is everything that happens to or through a protein. But this is obviously a very generic definition. So again, as you've heard last lecture, there are definitely very many different aspects to function, and it's generally a research field on its own. So how to really model the possible functions of proteins. The best we can do today is something called the gene ontology. It is so it is used by a lot of different peoples around the world. At its most basic level, it categorizes functions on 3 different levels. So first, the molecular function, the biologic processes of the protein, and finally where both of these two in the cell happen. So ideally, for every protein, you would have 3 types of annotations, each of them corresponding to one of these processes. And the whole gene ontology is organized in a hierarchical structure, which I will show you in a second. So again, what you ideally want to have is what we would want to have for our predictions is you go from DNA to protein. So people have already done this for us. So from 4 nucleotides sequences to a 20 amino acid sequence, and then we want to know what does this amino acid sequence do. And in this case, let's just assume this protein is part of this complex. And if this was the case, you would have an annotation, for example, binding ADP and catalyzing it to ATP. This would be the molecular function ontology annotation, MFO. In which process does this all occur? So what is the general process this reaction occurs in? It is for the synthesis of ATP, biological process ontology. And where does all this happen? It is in the context of the F1 ATPase synthase complex, so together with different proteins in the membrane of the cell, and this would be then the cellular component ontology. Different example. This is now real. This is for human hemoglobin, so the carrier of the oxygen in your blood. Again, you have three different types of annotations, one for each ontology. They're all disjoint graphs here, so there are no connections between them. On the root, you have again the three ontologies, and then the first children are very basic, are on the very basic levels of biological regulation here. The cell, obviously, a cellular component. Molecular function, so antioxidant activity, catalytic activity, so basically everything that an enzyme does. And if you go down the hierarchy, you get more and more specific about what the protein does until, for example, until a leaf here is really the most specific description, and in this case, it is, for example, bicarbonate transport.
How do we derive the numbers, the Go numbers?
They're defined by Go itself. So there's a Go consortium, and they come up with the numbers. Yeah, it's true. So actually, this is a Go ID, it's called, and this is a Go term. But yeah, I guess this distinction won't be important for you, at least. Okay. The background of our approach is that that evolution likes to copy and paste. So this means even if you have speciation events for example, if you at the origin of a new species, much of the old species is transferred to the new species. And you have here different ATPase proteins on the left from various different organisms. And you can see here on the right, they are all very similar in sequence, not exactly the same. So there are different, different mutations eventually led to different protein sequences, but as you can see here, they're all quite similar. And what our approach for this was that if you would imagine that you have just sequenced a new species, but you know the functions of for example this protein right here, you would simply look it up. You would look up the functions of this protein and simply say that your new protein is so similar to this one that we already know something about that it has the same function. And an intermediate question for you: so what is the name of this? What do you call this representation of sequences? Do you know?
An alignment.
Alignment, exactly. Is everybody familiar with this term? No. How do you know?
For the prediction.
Ah, you were there. Okay. I asked earlier who was there and nobody was. All right. So this is really basic stuff. So if you don't know what an alignment is, you should really read up on this at best. So right after this.
What's the terminology that we have to understand?
Alignment. Alignment, yeah. It's simply let's just say this wouldn't be ordered so nicely in blocks. So an alignment basically means that you find the ideal overlay of two sequences. So the best matching between two sequences, in order to really see how similar they actually are. Okay. So obviously finding so in this case it is pretty obvious, but so there are cases, there are pairwise comparisons that are not as obvious, and as you can imagine, so if you have one sequence with around 1,000 amino acids and you want to search it through a database with billions of amino acids, you will get into some sort of computational trouble. And for that, there are, so for string searching in general, there are quite efficient algorithms. One of the best is SciBlast, which does the search very quickly and gives you quite accurate alignments in the end. So the first thing that the students were supposed to do is, like, they take proteins that are not annotated, they SciBlast it against the part of a database called Swissprot that is already annotated. So Swissprot is a subpart of the database that I've just shown you. That is basically that has the most reliable annotations of proteins known today. So all the annotations of proteins in Swissprot are checked by human curators. And if they find a close match, a homolog in this database, they simply transfer the annotation of this homolog to the one to the protein that we want to predict. And there are different variants to this. So now we go into a little bit more machine learning oriented terminology. So you have something like a 1 nearest neighbor. So you only take the first homolog that you get back, so the most similar one, according to the e-value. The e-value is simply the chance that you find a match of this quality by chance. You could generalize this concept a little bit to the k nearest neighbor. So you not only consider the first hit, but the first k hits, the first 5 hits, for example, and you somehow compile the bag of functions that you get into your prediction. And you could even go further with weighted k nearest neighbors. So this means, for example, that you score the hits that you get by the chance that you got this hit randomly, and then again weigh every match, every go term for every protein that you get by this e-value, for example. Okay. Any questions about this so far? Is this clear to everybody? Okay, so the students had, 2 months, I believe, time, and in the end, we got a few methods back. So this was the first group: Ariane Böhm, Tatjana Braun, Rebecca Kastner, Cedric Landerer, Yannick Malich. Their method basically took the first 6 hits of a SciBlast query and compiled a score for each go term that they got. So they considered in how many of this first 6 hits a particular go term occurred. They predicted exactly 3 functions for each ontology, and they only used 3 different scores. So why exactly 3 scores? Why 3 terms per ontology? I don't know, but it turned out to work quite well, actually. So the basic concept of their method, again, is you get, for example, 6 blast hits for each query that you do, and if a go term of a protein function occurs in every one of the 6 protein, it gets a score of 1.0. If it occurs in any less than 6, so for example 2, 3, anything like that, it is assigned a score of 0.5 and a score of 0 in all other cases. You can then propagate the hits up the go hierarchy, and after they have done that, they reduce the tree a little bit to remove the most similar functions and get a sort of non-redundant output in the end. The second group, Mark Heron, Thomas Hopp, Stephanie Kaufmann, Dennis Kompers, Stefan Seemeyer, they use a weighted k nearest neighbor. So the k is actually can be chosen freely. They have quite a sophisticated continuous scoring scheme that I will explain in a second. That is based on the hit count, on the e-value of the hits, and on the overall structure of the ontology. They normalize the scores of the go terms they get, and they optimize all the free parameters in their method. And here's how the method works as an overview. So again, you have your protein hits 1 to n. So n proteins are similar to your query according to the blast e-value. The first score that you calculate reflects how good the overall blast result is. So how many good results did you get in the end? So for example, you could have one query where you get many good results, many very similar proteins, and another query where you don't get any or only one. This is sort of reflected in the template quality score. So you first take the logarithm of all the e-values, you build an average over them and the standard deviation, and take the standard deviation, and you do this for a couple of proteins, and you will, if you then plot the distribution of these scores, you get a Gaussian curve like this. And if you then predict a new query, you can look up in which percentile range the quality score of your new query falls, and then then can convert this unnormalized score into a range from 0 to 1, which is basically your which can be handled in a much better way in the end. And of course, besides the overall template quality score, you also need to score each Go term that you get. So you can associate each Go term of your results with the e-values of the respective results. So Go term 4 here occurs here and here, has e-values 10 to the power of minus 20 and 10 to the power of minus 3. So you can associate this term with the logarithms of these e-values. You do that for all the terms in your result. You take the maximum score and divide all the scores by this maximum score, which automatically turns these scores again into a range from 0 to 1. You then finally correct your predictions by taking the average over the whole path in the ontology, and finally, last step, you simply multiply the raw template score by the so-called combined leaf score to get your final score for this query protein, which is due to this template quality score, then also comparable to the predictions of other proteins. Last method, group C, Dominik Achten, Florian Auer, Maximilian Hecht, Peter Hönig-Schmidt, Michael Kieding, Manfred Roos, has similar features as the method that you just heard. So again, a weighted e-value based nearest neighbor, a custom scoring scheme that is continuous. And on detailed level, however, the methods are quite different. So they are actually not looking at the e-values or at the chance that you get this randomly. So, but on the positive identities. So this is very similar to sequence identity, and sequence identity basically is the fraction of amino acids that is identical between both proteins. So you calculate in this method scores, 2 scores. Each score is specific for each Go term, so there is no overall template quality score. First of all, you associate each Go term with the best sequence identity that you got in your whole result here on the left. And secondly, you also count how often a Go term appeared in your result. So for example, you could imagine that this Go term here appeared twice among all the proteins that you got back, that another Go term appeared 3 times or only once. And if you then divide all the scores by the full number of hits that you could have, so by the maximum number of hits, so by the hits that you got in your overall result, you again normalize it to a range of from 0 to 1, just as you did with the sequence identity, and in the end you again multiply the two scores to get a final score back. Okay, any questions about these methods so far?
So are there any tests about the...
Yeah, we will cover that very extensively. Testing. So how would you test how well your prediction works? So let's say we have protein A. We have a known experimental annotations that we did not use for the prediction, and we have a prediction from one of the methods that I just explained. So this prediction method would have associated each Go term here depicted by a node with a score between 0 and 1. So naturally, the more general terms would be predicted more reliably because they're easier to predict. So you have high scores at the top and rather lower scores at the bottom. You have leaf terms here for leaves and actually predicted terms. So we can ignore the leaf terms here. So it's a different story. So let's just look at the predicted terms. And what you do is, as a first step, you take only the most reliable predicted Go terms. So for example, you take the threshold at 0.8, which would leave you with two predictions, namely this term here and this term. And then you compare this prediction to the gold standard here. And in this case, you would see, so this term here is predicted correctly. That's a true positive. This term here on the left, however, is missing over there, so that's a false positive. And you have missed quite a few Go terms that would have actually been correct. So this entire part here is missing in your prediction. So these are false negatives. And with true positives, false positives, and false negatives, you can calculate a so-called recall precision curve. So recall is the fraction of proteins of Go terms that you covered. So in this case, you have covered one correct Go term out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So 1 by 11 is your recall, so around 10% recall, and you have made two predictions, and one of the predictions was right. This means we have a precision of 1 by 2 of 50%. So in this particular example, your point in the recall precision curve would be at 1 recall and 50% precision. Then in order to get the whole curve, you then lower the score for example, from 0.8 to 0.7, and you repeat the entire procedure. You again calculate recall and precision, and you have your next point. And you do that for every possible threshold here, for around 100 targets. So you get different recall precision curves for each protein, and in the end, you simply take the average. And this curve then here gives you quite a good idea of how your method does for the average query. Is that clear? Yeah. Okay. And finally, often curves are quite difficult to handle. So what you would want to have is like one number that tells you how good your method is, and so one way to boil down a recall precision curve to one number is by taking the so-called maximum F1 score. This is basically one point in your curve which has a very nice sort of harmonic mean. Yeah. Yeah. All right, so now we know how we could hypothetically evaluate our methods. And let's see. Yeah, let's do that and then stop. So what we did to really evaluate our methods practically is we took 10,000 proteins from the SwissProt database that were already annotated with Go terms. Templates was the rest of SwissProt, so around 400,000 proteins, and then we simply said use this part of SwissProt, the rest of the SwissProt, to predict these 10,000 targets that we chose, and pretend we don't know the annotations for these 10,000 targets. So, and we evaluated the methods in two different ways. So we gave methods a chance to only predict a few proteins. So to really make the decision, do I actually want to make the prediction or do I want to abstain from the prediction? In this case, though, this is the second part here actually. So a comparison only on the proteins for which the methods have actually made predictions. This is one way of the evaluation, and the other one was really that we said we want to enforce the prediction of all the proteins that we have, and in this case, if a method had not done any prediction, it was penalized with a recall of 0.0 for the entire protein. And yeah, this is what we got out in the end. So recall precision curves, so quite good curves and quite surprising ones too, because you have one method C, which covers rather low recall regions, but with a very high precision. Then you have another method B. That covers high recall with rather lower precisions, and finally method A, the very simple one from the beginning, which did not cover a large area of recall, but the predictions that it made were quite accurate here. So here you can see the two green points. And also random, so this means randomly taken Go terms from the whole gene ontology and giving the score according to their frequency in the whole SwissProt database also could achieve already quite high accuracies, although for very low recall. So you have some terms, some Go terms which are basically present in every protein that there is. And yeah, so to summarize this part here, so the implementations were quite hasty, so 2 months for something like that is not too long. The nearest neighbor approaches work surprisingly well, but we will have to see about that. And I will talk about that next time. The technical details of your procedure, so whether it's a k nearest neighbor or 1 nearest neighbor, and how exactly you score, they do actually matter quite a bit, as you see here. And but there may be still quite a bit of room for improvement here, because already the random predictor does quite a good job.
What does the meta stands for?
This is a linear regression of both methods combined.
Okay.
Yeah. So you can imagine this, that you predict a protein with one method, and with the other method you get 2 lists of Go terms, and if both lists have the same Go term, then you have 2 scores basically, and if only one of the lists has the Go term, you give a score of 0.0 for the one that is not missing. Then you can encode each Go term with 2 numbers exactly, and then you can do a linear regression on top, and then you get out this. Okay, so that's it for today. Any questions general about that? So now is the time.
How long did it took to run the algorithm? I mean, the first one was, but the...
Yeah, so 10,000 blastrons is actually quite CPU heavy.
Yeah.
But we have a 600 CPU cluster here, and this made things a lot easier. So this 3 days around for all poly methods. Okay, then thank you very much.